The participants are requested to kindly mute themselves and also off your cameras. We'll let you know after the session. Then you can unmute yourself for the queries. Good afternoon, everyone. I, Dr. Ravneet from Amity Institute of Biotechnology and one of the organizer of this FDP and the Master of Ceremony for today's session, extend a warm welcome to all of you to day 2 of this 5-day faculty development program on emerging trends in research, writing, and publication. Along with my co-organizers, Dr. Surya Pratap Singh and Dr. Arpit Srivastav, I'm delighted to have you all with us once again. Along with my co-organizers, I extend a heartfelt welcome to all of you. This FTP is being organized by the Amity Institute of Biotechnology in association with AIU, AUR, AATC, Association of Indian Universities, Amity University, Rajasthan, Academic and Administrative Development Center. We gratefully acknowledge the support of AIU, AUR, AATC in facilitating this academic initiative. We are truly delighted to have participation from more than 382 participants across the globe, making this FGP a vibrant and engaging academic platform. It's also a matter of great pride that we have esteemed international and national speakers joining us, bringing global perspectives to our discussions as this program aims to explore contemporary trends in research writing, ethical publication practices, and the role of emerging technologies such as AI in academia. We now begin our first session on global trends in research and scientific publishing. So before inviting Dr. Tiasha, I would like to briefly introduce our esteemed international expert, Dr. Tiasha. Dr. Tiasha is associated with Ton Duc Thang University, Vietnam, and is actively engaged in interdisciplinary research with strong international collaborations. Dr. Tiasha is globally recognized researcher and academic strategist with over 8 years of experience across India, Ethiopia, and Vietnam. Currently a researcher at Ton Duc Thang University, Vietnam, and an editorial board member for Scientific Reports, Nature Portfolio, she stands at the forefront of high-impact scientific publishing. Dr. Tiasha has established a formidable research footprint with a high H index and a consistent record of publication in top-tier high-impact journals, including Chemosphere, Journal of Hydrology, Journal of Cleaner Production, Scientific Reports, Results in Engineering, and the Journal of Big Data. Notably, one of her landmark studies was recognized as the most cited paper for 3 consecutive years in the Journal of Hydrology, underscoring her ability to produce research that defines and leads its field. With a PhD specializing in AI-driven water quality modeling, Dr. Tiasha's work bridges the gap between environmental engineering and advanced data science. A recipient of the National Energy Globe Award 2018, she combines her experience as a prolific first author and a journal editor to provide faculty members with invaluable insights into mastering the publication lifestyle, from manuscript drafting to navigating the peer review process of Q1 journals. With these few words, I would now request Dr. Tiasha to kindly deliver her session.
Thank you very much for your kind introduction.
Ma'am, we are delighted to have you with us today.
Thank you very much, ma'am, giving me after lunch timing. So I hope I can keep everyone awake. So that will be one of my prime objective. So
Sure.
let me share my slides first.
Yeah, sure.
Is it visible, ma'am?
Not yet, ma'am.
Now, ma'am?
Yes, ma'am. Now it's visible.
Okay, thank you very much. I would like to thank the organizing committee for giving me this opportunity to share my experience in the field of emerging research writing and publication, and which has been increasing and a global trend all over the world that research and only academics has not been highlighted yet, but more research concentrated work has been done over the years. And I especially thank Dr. Surya sir who introduced me to this program. So thank you very much. So my topic is from data to discovery. The advanced strategies for methodology visualization, high impact result presentation for Q1 level research publication. Before starting, I would like to share my academic journey. I'm Master in Environmental Engineering from Rajasthan during 2014 and later joined as Assistant Professor in Purnima Group of Institute in Jaipur. During 2015, I got an opportunity to join a United Nations program for underdeveloped countries to accelerate their uh, academic work, where I joined Ambo University, Ethiopia in Africa. I also there worked as a lot of research coordinators, in which there was one in COWASH and IoT programs funded jointly with Ethiopian as well as Finnish government, and then collaborated with multiple international seminars and fundings and industrial linkage. One of my research project was awarded as a national award from Austria. I finished my PhD in 2023 from Ton That Thang University, Ho Chi Minh City, Vietnam. Later, I joined in Dehradun during 2023 to 25, where I worked also as a research head for to proliferate and increase the ability of the faculties to produce high-level research as well as publications. Later, in 2025, I joined as an editorial board member in Scientific Report. And from then onwards, I'm as a visiting researcher in Tondaktan University. And recently, I joined as postdoc in UTM Malaysia, as well as International University in Malaysia as a research fellow. So this is my academic journey. So let's start with today's major events program for this. So our focus is how to make chart to design how to architect a visual narrative. So now you see just a histogram, but the target of today's lecture is to convert into more architecture narrative where as a researcher we can present our work like a storytelling and more revealing informations. So this is a graph I'm sharing. From these, the figures I'm going to share today is all with Q1 journals. So this is a scientific data journals report. As you can see the difference from just a chart to a new dimensional visualization techniques where multiple information have been shared. The second example I'm showing, this is a figure from my own research paper, Cleaner Production, where you can see a multiple information or more complex visualization graphs have been created. From during last 10 years, the visualization sector has improved considerably considering the different softwares and programming codes have been available or accessible to the researchers where the data can be visualized or presented in different variations, and more information can be gathered based on the domain expert ability to explore. So these are the just example, and this is the focus of today's FTP program to how to create from chart to a visual narrative. So there is why visual rules in Q1 journals. Why Q1 journals focus more in visuals. So we have 3 points here. First is the 10-second rule. When an editor or a reviewer sees your work, what in first 10 seconds what is the impression they get, and that will be the first is the your visual works, the figures, the charts which we have presented in our work. They are not going to read the whole paper, but the 10-second rule says that they are first going to skim through the figures. The second is cognitive load theory, which says we should reduce the reader's effort. When we are doing a literature survey, we come across multiple research papers. Whenever we are writing a paper, maybe we are reading 30, 40 different papers, and it's not possible for a reader to go through all. So if your research paper have more constructive figures, more informative figures, then you will able to reduce the reader's effort. Third is the graphical abstract revolution. Fourth, video abstracts, and fifth is social media engagement. So let's dive in with the each of the points, that how these are very important to understand that why keyword journals have more emphasis in the quality of the visuals. Here are two figures. If you are a reviewer or if you are an editor, which of these figures you will like more? Or which of the figures will give you more information? The first one or the second one? The answer is very much known. Definitely the second one. The second one gives you more information. The first one figure I have taken from a Q4 journal, whereas the second was is Q1 journal, Communication, Earth and Environment. So from the figure itself, as an editor, you can identify the work pattern, the objective, and how transparent the research work has been done. This is a figure from my own research work from Q1 Journal Cleaner Production. As you can see, the first one is a chord diagram. I hope you are familiar with these kind of different graphs and patterns which have been used recently. The second is sunburst diagram, and third is the common general bar graph. So these information from the single figure, the reader can have lot of information without going through the all the hard work of reading the text. So the first, the 10-second rule, and second is reducing the load of reading. Third is the graphical abstract revolution. As you may have noticed, in recent years, all the Q1 journals ask for uploading the graphical abstract. What is graphical abstract, and what are the the contents you should discuss? Graphical abstract should mirror the original text abstract. It doesn't mean that you just give something or just methodology part, but graphical abstract should be detailed as much as the original abstract what you have written. So let me give you an example. This is a graphical abstract published. What do you get? What kind of information do you get from this figure? Can you tell anything about the research? So this is a Q3 2019 Journal of Chemistry. This is a MDPI journal. Now let me give you one more example. This is another graphical abstract published in Q3 2018 Journal of Processes. So what kind of information are you getting? Can you understand the thematic area of the research published in this paper? Can you understand the result orientation? I said no. Let me give you one more example. So now you see, this is the Lancet paper, Journal of Infectious Disease. Can you see the difference in the graphical abstract? How detailed the information is. As a reader, when I see a graphical abstract, I can understand that what the research is about, what the objective is of this research is, and how the author is conveying the information to a more confined yet detailed information in a single picture. Let me give you example of another. This is a nature paper, uh, also showing the graphical abstract with detailed figures that what the research is doing. So this is a nature-inspired material-related paper. Another example is from my own research paper. This is a research paper of uh, chemosphere, and here you can see that how the research methodology is going from a wastewater and application of different clay and how it has been evolved to prediction models and input variables. This is my recent paper just accepted last week. This one also shows that how in Bihar region the water quality, meteorological data has changed, and this is a climate related paper of trend analysis. From the figure itself, you can analyze the study area, so you can easily understand that this is the study area is Bihar, and what kind of data we have taken, like 37 years of data from NASA, and what kind of analysis have been done. So it's very important that graphical abstract mirrors the original abstract what you are presenting. So these are the few things which will help to pass the first preliminary checkpoint if you are sending your research paper in a Q1 journal, and when editor or reviewer is checking your paper, so this graphical abstract and the visualization will have the first impact among the editors. So next is video abstracts. Now the next generation to the graphical abstracts are video abstracts. They have more impact, as more than 7.7-fold impacts, retweets, and article visits. So if we are thinking of that after the publication, how we can reach our readers, the research doesn't end in your lab, or the research doesn't end on your computer itself. It should reach to the readers. So video abstract and social media engagement is the most powerful way to reach to your readers. So how will you reach the reader? With the effective and powerful application of visual importance of today's methodology and result. So let me give you one example. This I have taken a snapshot of a Google Scholar of Dr. Zaha Mundir. He is top 2% world scientist, and this is his most cited paper, a survey on river water quality modeling using artificial intelligence model 2000 to 2020. And as you can see, 735 citations are there. So let me project you with his H index. Sorry. Okay, as you can see, he have an extensive work on research and very high H index. So let me now tell you the second part, that he was my supervisor in my PhD. So let me share my Google Scholar. This was my paper in my PhD time, my first review paper for my PhD work. You can see that being the 2% scientist, still my paper is the most cited paper. Why? As I told you, your paper doesn't end just in the journal or on your computer. It should reach to your readers. So what are the factors you should consider? The journal where this paper was published is Journal of Hydrology, and this was a subscription journal. Still, the citation was more than I expected. But why? There are a lot of reasons when you are publishing in Q1 journal. First, the quality of your work. Anything, any visualization or anything cannot overcome the quality of your work. Certainly, the quality of work is very important. So in this paper, I have reviewed more than 300 papers. Second is the topic you have chosen. The topic should be relevant to recent research trend. Third is the journal you are selecting. not just a Q1 journal, but the top or the most reader-friendly journals. So Journal of Hydrology is one of my research areas, top research areas. And then you should have a better presentation of your visualization, or every work should be presented so reader-friendly that it gets more readers and citation. So let's continue to the next. This is what is graphics. When you are choosing to design your work in form of graphics, you should have used to know your audience, use a proper color format, align your elements, prioritize your parts, highlight your work, and invest in figures, and choose your charts carefully. Your figures, your charts, and diagrams should be very prompt, presenting your real work. Just using any graph is not enough. The graph should have a in-depth information and knowledge that what your work really want to present or reveal. I hope everybody is still with me. So let's start with the basic phase 1, visualizing the methodology. I have given two examples. One, integrating data-driven prediction traffic resilience framework. This is a basic flow chart of a methodology from a research paper. Second is also a methodology presentation of research work. What do you think with this? Are you getting all the information required to understand the methodology of these research work? I would say no. So this is a Q2 journal, Applied Science, and this is a Q3 journal, Energies. Why I'm showing you examples that you can differentiate the evolution of all the diagrams and what kind of information a journal expects you to reveal when you are publishing in high impact journals. So let's go to a Q1 level journal. Sorry, one more example is there. Q3 atmosphere. You can see very simple diagram with less information. These kind of figures will get you rejected in the first stage itself. Sorry. So this is the first figure. Second one, this is a detailed information about the steps they have taken. All these are Q1 based journals. And this one, the last one, is my own paper, published in Marine Sciences, Q1 journal. And I would like to discuss, as you can see, from the start to end of the model checking, all the details have been given. If you are just reading the figure itself, you can understand that which models have been used, how the data has been combined, and then trained, and what kind of different splitting techniques have been applied, and what kind of data test have been evaluated. So these type of figures should be designed to get a visualization that what methodology have been applied in a research paper. So when we are working on the flow charts, we should have four things in the mind. First is moving beyond text. So we can use different kind of softwares like BioRender, Lucid Charts, or Lattice integrated TXZs. These are very excellent softwares which have professional figures, icons, which you will not get in Googling the in a search engine. They have very professional, different climatological, biological, or any other research field you can say, specifically biology, I would say. BioRender have very detailed figure charts which can help you to create a vivid information about your research work. Second is the pipeline view. Create a snapshot of your method so the reader can easily read your work. Third is consistency is the king. When you are working in a collaborative work, you may have more than 2, 3, or 5 researchers. But when, as an editor, I'm reading, I should have an illusion that this paper have been written by a single person. The consistency of the language, consistency of the headings and figures should be so well merged, so well written that one should understand that this research have been done by one person. The icon should be very clearly presented. The color mappings should be very clear and consistent. We should not have lot of color or beautification, and the meaning of the visualization will be ruined. And using semantic codings. So this is very important. And tools of the trade. So you can use different kind of tools of this for designing this kind of visualization, including the PowerPoint, Canva, Excalidraw, and Flaticons. I have used a few of them already, and they are excellent tools. And you can get a lot of experience using different kind of softwares together, and you need to edit them based on your expected result. So the next phase is next phase is advanced data visualization strategies. What do you see in this figure? The first is a bar chart, second is a line graph, and third is very colorful pie chart. What kind of information are you getting from this? Less informative? Very simple. Reduced analysis, reduced precision, and misleading. Very simple, or these kind of visual narratives don't give the full access to the reader the information required to understand the research or replicate them. So we have to go beyond the charts. This is a violin plot. This is from my own paper. This was used to analyze the error diagrams for different models. Second is Tyler diagram. This is the Journal of Environmental and Management. And this is a heat diagram from another paper. So as you can see, the color chart is very much important. These are the very popular these days applied for visualization. For heat maps, there are a lot of different applications. Heat maps only is not used like this, but you can use this for different kind of climatic change or weather reports. For example, these kind of world maps also can be used for heat maps. So these are few of the examples, and in the in the notes, I have already given all the details of the journals I have used for these figures. So these are the differences when you see from very good quality paper to the average quality of the papers. Next is advanced data visualization strategies. Second phase is tables and charts. So if I say give you a few minutes and tell you to ask you to tell me that what kind of information can you gather from these two tables. So what will be your idea? Maybe you need a magnifying glass to read these because they are very, very small. You have to zoom in a lot. And after a few minutes, you will be bored. And maybe some of you will sleep on it. What we do is use of multi-panel figures. Multi-panel figures are figures which have multiple information merged together. This is first figure is Lancet Digital Health. You can see a lot of information have been given in a single figure, and it's very easier to identify that what the figure is trying to reveal. The second figure is from result of engineering, where two river flow has been analyzed together in a single figure using different techniques. The third figure is frontier of remote sensing. This also shows different heat maps of a same study area. So these are multi-panel figures. When you want to assess a lot of information together, rather than have a simple figures, you should merge them together to create a more impactful or informative information altogether. Next is discovery factor, the power of caption. This is my first example where a figure is given like the patterns 1, 2, 3, D, E, like that has been given. So not a lot of information have been provided. Like from the figure itself, you cannot say what is this about. Next is another figure when inclusion of dichloromethane and chloroform and something, figure 1 to D. Again, very less information have been given in the caption area. This is a Q4 chemistry paper and Q3, the state variable profile. So these are the captions which are not welcomed anymore. So let me go to the better example. This is a Q1 Nature paper, published paper. I think its impact factor itself is 20 or something. So the figure had main is total and transboundary co-benefit associated with different climate scenarios. The figure itself is very detailed, and by the figure's caption, you can get all the information you need. You don't need to go for searching what is SSP, or you don't need to find what is RCP, because it's a compact with very high quality visualization with detailed caption. So it's very important that the caption should be detailed if you want to publish in high impact journals. Next is the narrative arc. A good paper should have the figure caption related to the objective. Here is the objective. The study aimed to investigate the cyanobacterial species, and the figure itself is very detailed, and I was not able to copy the figure ABCD detail because there is very less space in the slide, but they have given a detailed information similar to the previous slide about ABCD. So the narrative arc is that the objective should align with the figure's information. When I'm reading the objective, I should feel the objective has been satisfied or results have been very aligned to the given aims and objective. Next is contrast and focus. What do you see in this figure? For discovery of your work or good presentation, contrast and focus is very important. As you can see in this figure, encoding too much information, the color coding is wrong. Can you identify any of the areas from this? No. All the figures, all the dots or the colors have been chosen by the researchers are very similar. It's difficult for anybody to find that what information have been given in this figure. So this is a bad example of figure colors. Next, here I think all the colors have been used. These are called rainbow colors. So what information this rainbow color gives? Only beautification. No purpose, only distraction. If your color coding is giving information, then only it is useful. Just making it beautiful is not the objective of the researcher. So, next example is which data values are larger or smaller. The next figure you can see, you cannot identify which figure is giving the information of larger values or difference between the data values. Again, using rainbow data where the data cannot be identified based on the values is not a good example. So this is a good example of color coding or contrast and focus. Here, as you can see, the figure is very clear. All the information have been very clearly coded. You can identify any kind of bacterial information from the genus, as well as the figure caption is also very much clear. So this is a very good example of contrast and focus of a figure. Next is oh, sorry. Okay, sorry. Next is technical specs and ethics. When designing a figure, we should be very careful that we should uh, work within the ethics of our work. First is image integrity, avoiding beautification. We should always encode our images based on the ethical guidelines for cropping and brightness and contrast, resolution format, and AI in visualization. This AI in visualization is very sensitive topic. Role of generative AI in diagrams have been used very frequently these days, but now all the QN journals want you to disclose the application or use of AI in your figures or diagrams. So AI should be used only as a part of your help. It's just helping you as a tool. You should always able to edit it. And then how to you can utilize it based on the visualization technique. The main question is that who is the author? Keep in mind that AI is not the author. That's why the final figure generated by you should have the final touch, editable qualities. Second is the integrity test. The integrity test is that your figure should be based on original research, not totally AI composed. And the last is tool trap. Don't make so much use of AI or tools that the real result is hidden behind the figure itself. I have given some examples of different major journals or publishers, I would say, that who allow what kind of AI application of different fields. Like Nature Portfolio permitted partial or MDPI use permitted as assistant tools or elsewhere provide data manipulation and different. So you should before publication see that what kind of AI application is allowed for visualization itself. I've given some of the tools which you can utilize for designing your visualization. If you are using interactive visualization, Microsoft Power BI, Tableau, these are very excellent tools. If you are presenting in infographic or presentation tool, Canva, Visme, these are excellent tools. Specified qualitative research tool is Jiffy, Vosphere is very good for designing bibliographic review or designing the co-author charts. And fourth, which we or me, I self use is R Studio and Python because these allow you flexibility. They allow you editing and all kind of different figure, you know, like the size of the bubbles or designings and data information, everything can be very flexibly used if you are using code based R or Python. And I have also added some of the most popular libraries which can be used for visualization. For myself, I popularly use R because it's a free software, and ggplot or ggplotty we have used repeatedly for many visualization, and they work excellently, and they have detailed figure editing options. Editable files are generated. So we are in the end of our talk. So what kind of questions we have in mind after today's presentation? Why visualization is very important and why proper ethics of visualization important. Like if I gave you exactly 30 seconds to look at a paper's figure and without reading the abstract, could you tell me the code discovery? If no, then is it a Q1 paper? If you are not able to identify the research objectives from the paper figures, that means it is highly doubtful that it's a Q1 paper. Or if any of you have gone through that, when your paper get rejection or revision because your methodology is unclear. These are the questions for today's to think about. When you look at a methodology diagram, can you identify where the data has entered, where is the intelligence applied, or you have to search through the all the diagram left and right to find out the information. If you have to go forth and come back again, or you have to search for small details which are not given in caption, that means your figure is not proper or failing it. You're not designed your figure onto the best level of the expected figure from Q1 expected journals. Next is the rainbow color maps, global temperature. They look vibrant, but can be color blind river distinguish. So we have to be very careful when we are selecting colors and why we are selecting colors. Just sake of beautification, we shouldn't select the colors. Next is when we visualize AI performances like R square, or we are finding some errors. Why do we often hide outliers? In your research, outliers can give a lot of information, specifically in my field of climate change and water quality. A sudden rainfall or sudden change in water quality may present a discovery that there was something different that day. There was a storm, or there is a sudden effluent discharge in the river which changed the water quality. So don't always hide the information. Always try to find more information using the visualization itself, and it can reveal very deeper meaning in your research discussion part. Sometimes what we think is that whatever objective we have set, we have described them, we have discussed them, and that is enough. But that's not all. When we are visualizing, we are able to identify the small differences or their changes in your whole data. And that is the key part of your discussion. If you're finding an outlier, you have to find out the reason. Is it an error? Is it a issue at that time? And identify will give your research work more meaning, and it will help you to work in high impact publications with more descriptive analysis. Next is, can the audience understand the research orientation by reading? Why you are not giving more information in the figure itself? Does adding 3D effect to your bar chart increase information, or just it a junk? And the last, after today's session, will you still be using a simple bar chart or pie chart? Or are you going to retire today after this session and find out the latest visualization softwares, the latest graphs, which can give more information or give life to your research work? Thank you for today's session, and I hope that we will not be a researcher who reports, but we will become the scholars who reveals about our research with help of high impact, high visualizations, and give our research to the more emphasis like a storytelling, which have not been done before. Thank you very much.
You so much, Tyasha ma'am. The session is open for question and answers. If anyone has any query related to high impact Q1 generals or anything related to research and publications.
One question is there from Dr. Viren.
Ma'am, there is one question.
Yes, please.
This is on GG plot.
Mhm.
That Dr. Tyasha, Dr. Virendra Kumar is asking that DG plot 2 will be viable for no coding researchers.
Yes, of course. These, when even I am not I am not from a field of computational science. My field is civil engineering, specialized in environmental engineering, and with small coding skills, you can easily use RStudio and ggplot or ggplot2 with help of easily available codes online. You can design any kind of visualization or diagrams. So please don't hesitate to explore these kind of tools, because we are not going to know till we try it. All the examples I have shared in this PPT, and I will be happy to share this PPT with the coordinator or organizer. They can share with you. All these libraries are excellent for those who are not from the field of computer science. They have very user friendly and very easy to use.
Anyone else? Any questions? Any queries? Thank you so much, Tyasha ma'am, for giving a detailed perspective on research and publishing in Q1 journals and high impact factor journals. We really appreciate your time and valuable contribution to this FDP. We'll now take a short tea break, and again we'll reassemble at 3:05 p.m.
Thank you, ma'am. Thank you very much. And any of the people who have attended, feel free to contact me if you have any further question via mail or WhatsApp. Thank you for your time, ma'am.
Tiyasha ma'am, sorry to disturb you once again. There is
Yes, ma'am.
one query again being asked in the chat box.
Sure,
That
ma'am.
please give information regarding the video abstracts.
Ma'am, what kind of information?
They
There
are
are
asking that please inform about the video abstracts. I think they just want to ask that how to make the video of the abstracts, or there is like some online.
actually, yeah. You have to. There are a lot of lectures in elsewhere where you have to send them the original paper, and then they help you to design the video abstracts. Like different pictures have been added, and your own. Some talks will be there, like how I'm talking to you and I'm giving the information of my research work. So these video abstracts are based on the like based on the journals which they. I'm happy to share some links later, that where you can find the video abstracts information, how to create them, or what are the basic fundamental contents of them. So you can contact me directly via mail. You can take from the organizer and I can mail you the links.
Yeah, thank you, ma'am. There is one more query that if we use graphical abstracts in a paper, then do we need to have its textual abstract too?
Yes, of course. Both are 100% mandatory. Many Q2, Q3 journal may not ask for graphical abstract, but whenever you are submitting a paper, abstract in text is star marked, and many Q1 journals they mark graphical abstract also star marked. That means 100% important. So some may even allow not to have graphical abstract, but text is 100% needed.
Thank
Any
you so
more?
much, ma'am.
No problem, ma'am. Any more questions are there in the text?
Uh,
Yeah, in the let me share in my in this group itself my
yes, ma'am. Please share in the group itself your email ID. So if anyone wants to contact you, they can
sure.
contact you directly. Yeah. Thank you, ma'am. Thank you so
Thank
much.
you very much, ma'am,
Thank you
and
so much, ma'am.
have a
It was a
great
great
day.
honor and a great pleasure for us
Okay.
to have with you us today. Thank you so
Same
much.
here, ma'am. Have a great day, and congratulations on successful FTP organization. Thank you,
Thank
ma'am.
you so much, ma'am. Thank you so much. You are all requested to kindly reassemble after 5 minutes.
Thank you so much.
Vinay, sir.
Yes, good afternoon.
Good afternoon, sir. I hope I'm audible.
Yeah, I can hear you.
Okay. Thank you so much, sir, for joining the session. And welcome back, everyone, after the refreshing tea break. So we now move to our next session by Professor Vinay Kumar, sir, which is on translational research and emerging biomedical innovations. So before inviting sir, I would like to briefly introduce him. Professor Vinay Kumar sir is a professor and head in Department of Molecular Biology and the head of Biomaterials and Tissue Engineering Lab at Savitha Medical College and Hospital, SIMATS, Chennai. Sir is a distinguished researcher specializing in biomaterials, tissue engineering, and environmental toxicology. He earned his PhD in biotechnology from the School of Biotechnology at Jawaharlal Nehru University in New Delhi. With extensive experience in both industry and academia, both in India and internationally, Professor Vinay has made significant contributions to biomedical innovations and translational research. Currently, in this role, he leads interdisciplinary research focused on the design and development of biocompatible materials, nanostructures, and hydrogels for clinical and regenerative applications. Additionally, he is also the Department of Molecular Biology and is actively involved in curriculum development, introducing innovative academic programs at the university level. Sir has successfully completed a project funded by the Science and Engineering Research Board, SERB, at the Indian Institute of Technology, Roorkee. His research achievements also include 5 patents for hydrogels and nanoemulsions intended for clinical use in humans. He has also conducted 3 animal trials on V-star rabbit, rat and rabbit models for his hydrogel formulations and has supervised 5 human clinical trials in the Department of Surgery, Dermatology and Community Medicine at Savitha Hospital. As a sought-after speaker, Professor Vinay has also delivered numerous plenary, keynotes and invited lectures, as well as chaired sessions at prestigious international conferences. His pioneering work continues to advance biomaterials research with a strong emphasis on clinical translational and improving human health. With these few words, I would now request Sir to kindly start the session.
Okay, thank you very much, ma'am. So I would like to thank the management and a very good afternoon to everyone here. So before starting this, ma'am, I would like to know, I was supposed to talk on AI and plagiarism, or it is on the translation medicine. So I have 2 PPTs, which I should start with.
Sir, you
Can you just go with the title? Yeah. What
yeah,
is
your
it?
title is Translation Research and Emerging Biomedical Innovations. But then, sir, it's your perspective, whatever you think that would currently fit in today's era where the audience, they can maximum utilize and they can learn the new aspects collaborating with the traditional ones.
Okay, thank you very much. So can I want to share my slides.
Yeah, sure, sir.
Yeah,
You can share
okay.
the screen.
Just give me 1 minute.
So you can share your PPT, you can share your screen.
Yeah. Okay, so I think you can see my slides now.
Uh,
Visible?
Not yet, sir.
Okay. Can
Yes,
you see?
sir.
Yeah.
Yes, sir. It's visible now.
So this is the first slide. I think you can see the first slide.
Yes, sir. The title slide, Smart 3D Hydrogel Patches and Scaffolds for Regenerative Applications.
Okay. Thank you very much. So actually, a very good afternoon. Actually, so today I'm actually giving this lecture from my home because we have a holiday today in the Tamil Nadu. So it's a Tamil New Year here. Okay, so okay, so myself, Dr. Vinay, and I'm working as a professor in the Department of Community Medicine, and we work on the mainly biomaterials and tissue engineering applications. So our task here, I'm working as a research professor, and my task here is to do the research, and we are mainly trying to integrate the research with the doctors and the PG and the UG students, that are the medical students we have. Okay, so okay. So we can change the price now. Okay, so before starting the lecture, just I would like to give a brief introduction what we are and what we do here. So we have a total of the 18 colleges in the Savita group, and I'm working in the Savita Medical College. So mainly we offer two courses, that's the UG and PG. So when we say UG means it's the MBBS courses, and then we say PG means we say the MD. So they are doing the super specialization. So mainly we have two groups of the faculty. One are those who are teaching them. So they are the academic faculty who are the MBBS doctors, MBBS, MD, and super specialized faculties. With the cadre from assistant professor to associate and professors as well. Second is our research department, that where we work. We are around 75 people here who work on the research and development. And our task is to teach the students, and also we mentor the students and faculties to help them in the doing research. So here we have these are classrooms and the hospital facilities here. We also run a few courses. Yeah. So we also run a few courses here. Just I would like to, yeah, we got the pointer here. So we also run a few courses like MD in different specialization, a few medical correlated master courses like we have MSc in anatomy, biomedical biochemistry, medical biomicrobiology, etc. A few fellowship also are offered at our college. Then this is our cell library, and this is our patent museum. So we have around more than 500 patented products for the medical and healthcare which have been placed in the museum. And a few are also available in the market, so that it can be helpful for the other people as well. So coming back to the my work, mainly I work on the biomaterials and tissue engineering. So we work on the developing various biomaterials, mainly the hydrogels and the nanopatches for the medical applications. So we have developed a few of the products for the last 3.5 years. I'm working here, and we have developed a few products like hydrogels. So there are a few properties that we see when we consider when we are preparing for a hydrogel. So hydrogel is quite common, and they are not only popular in medical industry, biomedical application, but they're also useful in the engineering applications. So in the biomedicals, they're used for the various applications, and there are different types. For example, we have hydrogel patches for the tissue engineering. Stimuli response hydrogels, these hydrogels, what they do is whenever there is change in the temperature or something, they turn from the liquid to the gel, and they are reversibly converted from gel to the liquid. So that helps in temperature regulation, etc. For example, if you make one hydrogel, that is a liquid at a room temperature, but once we're injecting in the patient, it turns into a gel. So that give us sustained and good drug release. Next is that we can have a injectable hydrogel system. So we have prepared a few injectable hydrogels. So injectable hydrogel, what they do is they are their consistency and the content is such that we're able to fill in a syringe, and we are generally injecting directly at the site of the injection. Or mainly we are using it for the arthritis and joint pains. Then we have regenerative applications, like we have a skin and wound healing, and then we are going for cartilage regeneration, bone tissue engineering. Then we also work on the biomed, we can say cardiac application, like for the myocardial infarction, heart attack. And we also have developed and a few patches which can apply on the cardiac. So a few animal studies are going on there. Then while developing a hydrogel, we have a few property, like we want to check whether what are the property like porosity is there. Porosity is mainly that because the hydrogel is mainly used for the we can say drug release property. So for the drug release, the hydrogel should have a high porosity. When we say porosity means, because pores should be mostly are the nanomaterials, the pores should be there, or you can say the pore number should be more, so that whenever a hydrogel is there and if the drug is applied to the hydrogel, it will able to bind maximum sites. Then a few second property is the ECM, that it's should be mimic the extracellular matrix, and so that the natural cells will be able to proliferate over it, and they can also migrate from one place to other. Next thing is that we say it a hydrogel. The reason being the hydrogel, because they have a major, one of the major property, main property we consider with the hydrogel is that it should have a higher water retention capacity. For example, if you talk about what is the one of the best and simplest example I give in the class is that we use the bandages. We have the bandages that we use whenever we get a wound or cut in the hand, and we are applying the bandage here. And after we applying, we feel that it has a property like it can absorb all of the moisture content. It's healing it, and also it's giving the antimicrobial activity. So because of this gels have the polymers which are able to absorb the maximum amount of the fluid from the wound. That's a we say, so it should have higher water retention capability. Secondly, next thing is the biocompatibility. Whenever you say biocompatible means whatever hydrogel, whatever the material we are preparing in our labs, they are going for the ultimately for the human applications. When you're going to apply in the human skin, if I say we are going to apply your bandage or nanogel on my skin here, what will happen here is if it's a cut is there, or we can say a wound is there, the gel is going to interact with the tissues and the blood vessel as well. So if our product is, you can say cellular nanoparticle or copper nanoparticles, they have the toxicity, some level of toxicity. If you are applying directly on the skin, this any of the toxic, we can say biomaterial, it is going to kill the cells. So if it is going to kill the cells, it means your product is not a biocompatible. We say like, we can say like cytotoxicity related to the biocompatibility. So if a product is cytotoxic, it is going to kill the fibroblast cells, cardiac cells, or kidney cells, or any of the cells that are biomaterial have been targeted. Then it means your product is not a biocompatible. So generally we have some in vitro assays, like we take the whole human blood and we apply our biomaterial in there, and we check how much of the lysis of the RBCs is done by this. Next thing is very important here is that we say it's a controlled growth factor, or we can say controlled, I say simply say controlled drug release. If I say controlled drug release, it means, for example, in the case of a bandage or any of the hydrogel, if you are applying on the skin, when you apply the bandage, it stay for we can say 8 to 24 hours on the skin. It means it should have the property to release the drug over a time period slowly for 24 hours, so that it can have the drug efficacy is maintained over a prolonged time. So that is one of the important factors here. Okay, so preparing hydrogel, we use different polymers, so that can be synthetic and natural as well. So because synthetic polymers already we have, and they have some of the limitations like biocompatibility, toxicity also have been observed. So generally nowadays, what is preferred is called natural polymers, like we have a collagen, Collagen is it gives the cell property. It also mimic the ECM. Then gelatin is there, and it can be easily printed, and it can be also photochrons linked. Then alginate is there after the hyaluronic acid, chitosan, etc. So, if I ask you, most of them, at least everyone have heard about the chitosan because it's easily available. It can be extracted from the fish egg. It can be extracted from the crab cells and many other from chitin from the other shrimps also. So that's quite cheap and we can say useful and also biocompatible. So these are some of the natural polymers which are used for making the biopolymers, or we say hydrogel, and when we say it's a 3D printing bio inks. So for a 3D printing bio inks, it has some certain properties that we check before actually printing these materials. Okay, so next thing is that how actually biopolymers or how hydrogel is made. I'm not saying about a 3D hydrogel. We're not talking about that now. I'll say only the hydrogels. Hydrogel is something, a liquid that have a it's more viscous than a liquid or water, but it's less viscous than a very thick gel or a solid material. So to get that consistency, and that should be irreversible, we need to do some of the curing, or we can say it's the treatment to the chemicals. And one of them is the, here you can see the ionic cross-linking. So what we do here is we take some of the ions, like we have calcium. So here what we do, we take the biopolymers, like we have chitosan solution along with the, we can say we have polyvinyl alcohol, and you are mixing both the solutions, but it will not give you hydrogen. It will be only a liquid solution. Even if you take the any of the polymers, collagen, etc., it is not going to give you a hydrogen. It will be a solution only. So it will be a liquid flowy solution. So to make it a hydrogel, what we do, we take in the syringe and we add a dropwise in the calcium chloride solution. Once you are releasing the droplets, it's going to turn into a round hydrogel because of the hydrophobic interaction and many other interactions in there. Second thing is we also use here sodium hydroxide to make this cross-linking beads, hydrogels. Second is a thermal gelation. We say it's a thermoresponsive gel. Thermoresponsive gels are actually what happen. They are at room temperature, they are only a liquid solution. And once you are keeping them in a high temperature, say 37 degrees Celsius or 50 or 40 degrees, they turn into a gel. For example, one of the best example here is the carrageenan. And also we can use gelatin, agarose, etc. Another method is that what we do, we take the solution A and B, like chitosan and PVA, and some of the polymerizing agent, like we do in the SDS stage before running a protein through the gel. And after that, we keep it under the UV or visible light to make the cross linking, and it gives you a gel. Next is the enzymatic cross linking. And here also we use of certain enzymes to make the gels, to make it convert into a gel. And here in the physical or chemical cross-linking, also we use glutaraldehyde here. But the glutaraldehyde is a highly reactive cross-linking agent. And once you are just adding in there your polymer solution, immediately you make it a gel. And so, but when we check this glutaraldehyde cross-linked gel in the cell lines, fibroblast and keratinocytes, we found that it has a very high toxicity, and we are not using that one for now. And also we try to reduce the glutaraldehyde concentration, and we are working on optimizing that process. So next thing is what are the types of the patches we have? So what are the 3D patches we can make? We can make a 3D porous patches. Porous means because any of the materials, what we generally do, we make the hydrogel and we're keeping, when we lyophilize it, after lyophilization is give a very spongy structure that have the holes or pores that can be observed in the scanning electron microscope. And we found that that is a 3D scaffold. Then we say sometimes it we say micro challenge scaffold, then cell laden what we do here in the cell laden bioprinted scaffold, we add our live cells. It can be cardiac, it can be fibroblasts, it can be keratinocyte, it can be bone cells or whatever, or cartilage cells along with the printing of the 3D scaffold. So that the live cells are also there and it can be used for the transplantation and the graft applications. After that, a few different type of hydrogel is the layered hydrogel. What we do here is we place one of the layer of the one polymer, then second layer, or maybe third layer, or maybe what we can do here is we can have one of the drug here. Second one, we can have a cells or any of the antimicrobial agent in the third layer. So depending on the target and specificity, this can be optimized. Okay, so we have a total of 8 3D printers, and 6 are used to make the dental implants, and we can use it for the preparing the titanium and all metal implants. And another 2 we have in our Savita Medical College here to make the biomedical specimens. So here on the left side, you can see these are the biomedical specimens which make to teach to the MBBS students for the different body parts. Second one here we have the extrusion bioprinter that we actually use and we have been using for a long time to prepare different hydrogels in our laboratory. Okay, so this is one of the specimen made by this 3D printer, specimen printer for the understanding of the brain. And we also have created for the muscle simulation here. Muscle, so because actually getting a cadaver for a biomedical, for a MBBS student practice is not easy. So we need to print them in the simulation using the 3D models to understand the anatomical structures. Also, we have printed here heart. Then vascular structure, like we have the veins and here is the arteries. After that, this is the upper limb that we have printed within 3D, and to make it a little bit more looking good, we can have a cover over it to look like more like a human. This is the lower limb simulation. So this is lower part of the leg, and apart from that, not only that, we are working on the translation medicine. So we have a medical college plus hospital. So it's a big hospital with a 1,600 bed capacity. And to make the inpatients, we have printed a specialized 3D support for one of the patient who was to give the support for walking. So that is also, so it means that these 3D models and the 3D we can say program can be modified to make specialized treatment products. Okay, so next is this is the gastric model that have been used to diagnose for the medical students, and these are the different vertebra, and yeah, these vertebrae, and this is the foot anatomy, so that have been printed. So for printing these materials, we have the bio inks, or we can say wire-like structures are coming. So so these different they have to do like these are mainly PLA and other additives are there which are used to make these specimens. Okay, so this is the ventricles of the brain using the which have been printed using a PLA. Okay, so that was about the 3D printer that we have. If you talk about me, what we work on there, so mainly our research work is focused on the producing the biomaterials for the actual clinical applications because we are working with the doctors and so publication we do and also we do research grants, but major part is that we have 30 super specialty departments in the hospital and I'm sitting mostly in the hospital. My room is there. So we are working with the doctors from the general medicine, then we have community medicine, anatomy, surgery, all the hospital departments that you experience while we go as a patient in the hospital. So our task is to make the product and help the doctors to develop a product or we can make the product and we can give to them for the human trials. So depending on the what material, what kind of hydrogel we are preparing, sometimes we prepare for different departments. For example, it can be a plant extract, it can be a we can say nanoparticles, nanoparticles, something that we're going to mix with the polymers. Like here we have used chitosan and the polyvinyl alcohol along with the lignin. Then we have some methods. Then we are making the hydrogel. Like here we have used sodium hydroxide to make the hydrogel. And once the hydrogel is made, we lyophilize it, freeze dry, and after freeze drying, we send it for the characterization like scanning electron microscopy to see the surface morphology, how the pores are formed or not. Then we also go for the transmission electron microscopy, then TGA, so to check the melting and all etc. behavior. Then we have FTR and XRD to know the functional group, size of the particles, and crystallinity, etc. After that, we go to the in vitro assays. In the in vitro assay, we have a set of defined parameters, like we can go for antioxidant activity, hemolytic assay, or antidiabetic assay. And if it's very specific to the immune markers or something, we can go for the anti-inflammatory activities as well. This is just one of the simple layout that we are following. After that, we go for the cell line activity. Like if it's a topical application, if we're going to apply on the skin, we can go with the fibroblast or the keratinocyte. And we check the toxicity along with the confluence of the cells, how many cells are killed by the material, at what concentration. It must be 10, it may be 10 microgram per ml. Sometimes it is more toxic. Even at 10 microgram also, some materials are even at 500 microgram per ml also, they are not toxic. And some of the products might have, we are planning for a cancer patient or something. Then we also check for the on the cancer cell lines. After that, we go with the animal study, and we define the population of the animals that we want to actually work on there. And then what we do is, depending on the study, we make the wound on the mice or rat, and we check the wound healing on the day by day basis for a week, 2 weeks. Okay, so these are a few of the hydrogels that we have prepared in my laboratory. So this is the first one I told you. Here, what we do here is, okay, so here what happened here is that we are taking the hydrogels and polymer solution in a syringe, and once you are dropping it drop wise, one by one from the syringe, what happened once it's going in the calcium chloride solution, or we can say, or it might be sodium hydroxide solution, immediately it turns to a hydrogel. And this is the final product. We wash with the distilled water 2 to 3 times to remove the NOH, or we can say calcium chloride. And after that, we can use for the in vitro assays, or you can lyophilize it for the characterization. Second is the, okay, so this second is the chemical cross linking method. Here, what we do is that we use here what we do, this is actually a liquid solution, and we are adding here dropwise the glutaraldehyde. Once we are adding the glutaraldehyde slowly, immediately it start forming the gel. After some time, within a minute, or you can say 30 seconds, it will turn into like this gel. So this is the fastest method, but the toxicity is higher here. This is the last one. This is the 3D method, that extrusion printer we have. And here we have to identify the material which is quite a fluid and which does not break while printing, and it should be it should be stable at room temperature. So this is the final output here. This is our 3D hydrogel in the form of the rectangle patch here. And here what we can do is we can optimize, or we can regulate the spacing between the gel, and also we can customize the layering, number of layers on this hydrogel. So this is part of the 3D. 3D, and after this one we can modify in the form of the, okay, so here this one we can modify the patch, and we can have if you want to have a rectangle, or we can say round shape, any of the shape we can make with the hydrogens. Okay, and this one is the we printed last time, we customized the software and we try to print the human nodes. So this is the human nodes that we printed using the carrageenan 1% polymer using our 3D extrusion printer. Okay, apart from that, we have made a few products, like we have made a chitosan using chitosan PV and apple extract, the gel for the wound dressing. And these all of the products are a thesis product of the MD students from the different departments. And the next one, we prepared the orange peel extract for the burn treatments. And we have you prepared the carrageenan, using carrageenan to make the injectable hydrogel. Actually, carrageenan is more important here because the carrageenan also have that thermostable property. And once we are increasing the temperature here to 37 degrees Celsius, it turn into the solution turn into a liquid gel here. Next is that one of my PhD students have been working last 1.5 years, and she prepared beta cyclodextrin inclusion complex using the ginger extract and flaxseed. And she is working on the ischemic attack and myocardial infarction. And we have working on the how to apply on the animal model and the human model as well. So this inclusion complex is ready. And we are going to we are thinking how can we make a patch to apply on the cardiac patients. And the third one, this is also we have made using the curcumin and cyclodextrin and MOF for imaging and diagnostic. And this is one of here we can see it's on the left is the perennial cooling gel. This is one of the PG from the OBG department. CHMA to apply this hydrogel on the patient with the C-section or cesarean surgery to make you can say provide a cooling after the surgery. This is one of the product made by the Department of Community Medicine. So all of these products are made by the PG under my guidance. So here is the herbal balm we have prepared for the insomnia cases by the one of the PG from the Department of Community Medicine. And this is the bacterial nanocellulose patch that have been made to apply on the spinal cord for the cardiac applications. Okay, and so next is this is sodium alginate hydrogel that we prepared for the post-surgical wounds using from the department of with the help of the department of the general surgery. And this one is for the diabetic foot ulcer. Actually, foot ulcer is one of the first area that I'm working on here on it, because the foot ulcer would happen in the diabetic patient. I'm not sure how many of you know about it. But for the diabetic foot ulcer, it's a very serious condition, medical condition, and who are suffering with, they have the wound, or we can say a hole in the foot of the patient that never heals. You can apply any of the antibiotic, you can give any of the drug, it will heal for some time and again reback. And sometimes the situation is such a dangerous, or you can say goes extreme, that we need to ampute the leg of the patient as well. So that one we have prepared for the diabetic foot ulcer dressing using the copper sulfate and chitosan. Then also we have used CMC here to make the gelatin membrane. So mainly for topical application in the pediatric wound care. CMC is the carboxymethyl cellulose, and carboxymethyl cellulose is actually when you make a solution from the carboxymethyl cellulose, immediately it turns cloudy and it feels like it made a gel. But it's not actually gel, it looks only suspension, we can say it's a cloudy suspension. So making hydrogel is a little bit challenging with the CMC. Okay, so these are some other products that we have prepared from the ginger, using the ginger extract and the PEG. And the flaxseed, this one is of the same. So one of the we can say intern student last year extracted chlorine E from the Spanish means is we say is the palak from there only for the photodynamic therapy, and we are also working on that. So and she also extracted the chlorophyll A. Okay, so this product we have made from the hydro. We actually one of the PG from the Dr. Suganya from the Department of Dermatology. She prepared this patch to apply on the nails of the patient who have the, we can say, damaged nails. And this one have was made using carrageenan, salicylic acid, and biotin. And the product have been this product have been patent have been filed for that, and she also completed her thesis successfully. So mainly these are used for the nail psoriasis and discoloration. So because in dermatology they got many patients whose nails have been damaged or the discolored. So for them it's quite useful product. Okay, so apart from that, we have animal house, and generally we do around 10 to 5 animal trials every month in our animal house. So we have also completed now 5 of the 6 animal trials. And what we do here is generally we do try to do using the wound healing or burn we can say burn trauma trials. What we do here generally we take we make one wound or remove the topical layer, and we apply the our hydrogel here to see the progression of the wound healing as compared to the control where we are not applying the hydrogel. So this one is actually polyethylene glycol and chitosan hydrogel that we prepared last time. And before applying actual gel, what we do, we also remove the hairs from the mice, and we see whether they got any allergy or any reaction before applying the gel. Okay, so here is the one of the PG have done a trial on the animal trial for their product, and she prepared one of the hydrogel using the lignin. So this is the lignin hydrogel that was tried on the 3 rats here, and also we have used one of the standard drug, that's a povidone. And we observed the changes in the wound healing, and we can say that the reduction is the size of the scar over a period of 13 to 14 days. And a significant, we can say that a significant wound healing, or you can say wound reduction was observed. So also you also completed the thesis now. And this product also have been filed, patent have been filed for this. Recently, we have also made one anti-repellent mosquito cream from the Department of Community Medicine PG, and she tried it on the rabbit, and she checked whether it has generally that it was a quite effective material, and we tried in the mosquitoes also. We have from the we have that one of the ICMR funded entomology laboratory. There we check the its efficacy of that anti-repellent cream, and then we checked on the rabbit also. Okay, so the workflow is that after in vitro go with the animal trial, then we go for the human trial as well. So this is one of the example how they do. Like for example, this is for the lignin hydrogel that CO used on the animal model, and what they do is they check the pain score, or we say it's a visual analog scale. What they do is they take the patients, and they apply the hydrogel. They ask to apply the hydrogel on them on their wounds, and they check whether there is a reduction in the pain in the patient. So here this is the scale we say pain score, and on the day 0, day 3, day 7, they observe whether there is reduction in the pain over a period. So here the we have taken the this is yellow one is our lignin hydrogel, and the second one is the standard drug, that's the povidone. Second thing is what the she checked is that she checked for the erythema, that's the redness and of the skin on the patients. And here also the first one is the our product and second one is the standard drug here. Okay, so this is the anti-mosquito repellent cream that we have tested in the entomology. Actually, this is the lab is funded by the ICMR and it is run by our VC, who was the previous ex-director from the ICMR, VCRC Pondicherry. Okay, so then we have human trials. So out of those, I have now around 17 PGs from different departments, of which 5 human trials have been completed, and these products also have been patent have been filed for them. And apart from that, I'm also the in charge for the flow cytometry laboratory, where we work on the detection of the leukemia and the cancer patients. Apart from that, we work on the different research like we can say stem cell studies and other studies also using the different markers. Okay, so these are my PhD students. So Kalpana is working in biopolymeric that is scaffold for the skin wound healing. Another one, Alawadai, is working on the alginate based hydrogel for the excision wound rat models. And Himavati is working on the cardiovascular disease and she developed the beta cyclodextrin, flaxseed, and ginger extract this inclusions. And then another student is working on the Gopika is working on the microplastic detection in the clinical samples. And recently we're trying to develop the SOP for the extraction of microplastic from the human whole blood, placenta, and also from the cord blood and meconium from the neonatology department. And Arif is working on the growth factor induced 3D patches from the burn surgery patients. So these I work on mostly on biopolymers and microplastic. Is also one of my area that I'm working for a long time. Okay, so these are the PG students who have been working on different projects from the different departments. Like we have dermatology, community medicine, then general surgery and OG. And 4 to 5 more have been added to the list that I have not included here. So they work on preparing the pimple patch for the skin, like this is from the dermatology. So she made one pimple patch to apply on the skin. Actually, many of the products already there in market. When you got a patch for mainly for the women's, so the patches try to heal faster as compared to when you're not applying the patch. Next is the customized nail thick to prevent discoloration. That is Dr. Suganya that I already have shown you. Lignin basis, that is same product that we use for the on the human patient, then mosquito repellent. And one of the PG also made the anti-scar cream for the dermal surgery and nanoemulsion for the insomnia. After that, this is also one of the nail liquor they have made for the different medical condition in the nails. Penile cooling gel, I already told that it is made by the Dr. Sri Devi from the OG department. And then we have also made from the aloe vera, one of the formulations for the topical application, mainly for the dermal applications by Dr. Divya from the Department of Dermatology. She completed the thesis and also this product have been patent have been filed. Then we have also made insulin infused ointment for the diabetic foot ulcer and Dhatura oil extracted cellular nanoparticle for wound repair. So mainly we work on if I summarize, I work on the nano hydrogels and we can say nano emulsion, nail liquid. We try to make it sustainable on the one side so that once the product is ready, when you're going to throw in the environment, it should not contaminate the environment and it should be biodegradable. Yeah, actually, I didn't tell about the biodegradability. So most of the materials hydrogels we prepare, we check and we check it is the biodegradability in the different solutions like PBC or PBS. or you can say distilled water as well. We check its drug kinetics and drug release profile as well. Okay, so these are the MBBS students who are working under me, and Karpaka Vinayakam is one of the students who is working on the spironolactone patch for the cardiac failure. Actually, spironolactone is one of the drugs that is used for the myocardial infarction or heart attack. And so he prepared using the bacterial nanocellulose, he prepared the patch, and we are still working on that. Another two students, they are working on the understanding the biomarkers from the flow cytometric biomarkers like CD3, CD16, 25 biomarkers for the hypertension and also in the diabetic 2 patients. Okay, so that's all, and thank you very much. So it's a quite interesting and engaging session here, and you can follow this is my LinkedIn QR code. You can follow me up here. So I hope the lecture will give at least a few things we could able to get in here that will help you for your ongoing research. And I would like to thank you again, the management, for giving me this opportunity. And I welcome the questions from the audience. Thank you very much.
Thank you, sir, for providing your insights on hydrogels and their biomedical applications. The forum is open for question and answers. Anyone has any query?
Hello, sir. Am I audible?
Yes,
Am
ma'am.
I audible?
Yeah, I can hear you. Please tell me.
Yeah. So I would like to know whether CMC, you told us that CMC can also be used for hydrogel formation, right? So
Yes.
what are the changes we should make in the CMC so that it can act as hydrogel?
Actually, what I say is that why you want to go with the CMC as only, because we have around 20 polymers that we have made and we can use it. So CMC is little bit its consistency is very thick and it makes like a cloudy solution. So what we the thing is that, for example, if you see any of the commercial formulation, any of the gel, like we have diclofenac gel, that for the pain, it's a transparent, you'll see that. So whenever we make any of the formulation, first step is to make the polymer solution or any of the solution transparent clear. And it can be only be transparent clear when it solubilized properly. And we need to actually check you can check for the pH, and pH is the major factor that plays the role. Second thing is we have to optimize the temperature. Temperature of the solution is the major factor there. So we can make CMC also, but it have been reported in a few of the studies that it can be useful. So we can do. But first thing is that is it really you want to go with that, because it's a little bit typical, but you can make it obviously.
Okay, sir.
Yeah.
So, sir, can we treat it by using bleaching agents to make it clear?
Using the bleaching agent for on what? On a CMC?
Yeah, yeah.
CMC, but I'm not sure what application are you planning for?
Just I'm thinking to work on some type of this aspect in which we can develop something from using CMC, or we are deriving the CMC from the waste. So I'm
Mhm.
asking in that aspect because I have not thought about any application right now, but
Okay.
I'm just discussing you about the possibility whether we can use it in that way only or not.
Bleaching, actually, I am not sure bleaching what purpose it's going to serve here. Because what we do is like we are in the hospital already, so we need know the application where we're going to apply it first. So we start from there, then we check with what are the products available in the market, and we compare them and we see what can be made. So CMC you can try, but bleaching I think is not a method there.
Okay,
Next,
okay.
polymerization is the first step. You have to see if you're going actually see. Thing is that we say hydrogel. Hydrogel, I told that it should have some consistency. We say that is little bit viscous. But it's
Okay.
not mandatory. You can to make a hydrogel. You can go with the solution also. We say it's a nano emulsion, and then we can say liquid also. Liquid is something like we use a nail paint. That is a liquid actually. So it's
Okay.
not mandatory to make a hydrogel. And the second thing is that maintaining the in vitro activity, or we can say activity at the application is very important. For example, you can make a hydrogel while making cross-linking or interacting with a different solution. It
Mhm.
loses some percentage of activity. So activity is the first thing that we should focus on.
Okay. Okay.
Yeah.
So I will contact you regarding this in the coming time.
Yeah, sure, sure. We can have a talk. We can discuss this, and sometime we can visit also because I do frequently workshop every year around 3 to 4. So that actually I did first workshop in 2023. So that we demonstrated the first beads, how we can make the beads from the hydrogel, because people have heard about the hydrogel but they don't know. So you can come and visit also. We can discuss about it. Sure.
Yes, sir. Yes, sir. Sure. 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 Sure.
Yeah.
Thank you, sir. Thank you.
Thank
Very
you.
nice lecture and very elaborated work on hydrogels.
Uh, Once again, thank you so much, sir, for your valuable contribution to this FDP and for providing your valuable insights. It was indeed very helpful for our faculty members. So at the end, I would also extend my heartfelt gratitude to our esteemed speakers, faculty members, research scholars, and all the participants. So there is one more question in the chat box from Dr. Vijay Chaudhary. She would like to know whether you are encapsulating cells in the tissue regeneration for diabetic foot.
Encapsulating for the diabetic food. Actually, when I say encapsulation, what happened here is that we are making mainly beads. So we don't do actually encapsulation. So mainly we do cross-link the hydrogels. So generally the task is that we have to apply it on the surface of the patient mainly. So it's a topical, mainly we prepare the topical hydrogel. So we don't need to encapsulate actually. So rather than encapsulation, what we do, we cross-link it. And we cross-link that one, the polymers. And while preparing the drug, while preparing the hydrogel itself, we add our drug. One method is that. Another method is once we made the hydrogel, we are incubating in the drug solution so that the drugs get bind over the hydrogel.
Okay, sir. Thank you so much. So with this, we come to the end of this day 2. And there is an important announcement to make that tomorrow's session will begin at 10 a.m. sharp, both in the offline as well as in the online. So tomorrow, the first session will be from 10 to 11, and the second session would be from 2 to 3 p.m. So all the online and offline participants are kindly requested to be on time. With this, we conclude today's session, and thank you so much, sir, once again.
Okay, thank you very much.
Thank you, sir.
Yeah.